All right, everyone. So now we're going to talk about the final part of basically this chapter, which is we're going to talk a lot about cellular respiration versus fermentation. So in cellular respiration, which we know can be both aerobic and anaerobic. So the difference between aerobic and anaerobic is what they use as your final electron acceptor. Aerobic uses oxygen, while anaerobic uses inorganic molecules. That's the difference. But the good thing about these is that cellular respiration, both aerobic and anaerobic, they reduce chemical compounds to NADH and FADH too. And they also create this electrochemical gradient. So this is basically where they're passing the electrons in these whole complexes in the mitochondria. They keep passing them around and they're creating a proton gradient in the intermembrane space. And those protons are then moving back through chemiosmosis to fuel the ATP synthase. Both of aerobic and anaerobic do it. The main difference between aerobic and anaerobic is basically the final electron transport, sorry, the electron acceptor. So if you go back here, so all of this happens within the aerobic and anaerobic, this passing of electrons between the electron carriers. But this is what is basically the only thing that differs. So in aerobic, oxygen is there. In anaerobic, an inorganic molecule is there. That's the difference there. When it comes to fermentation, fermentation does not use an electrochemical gradient. It does not. It does not go through the citric cycle and all these basically an electron transfer system to power ATP synthase. Fermentation only creates energy through substrate level phosphorylation. So this is basically through glycolysis, and then basically donating the electrons from from NADH to a organic molecule. So here in fermentation, the final electron acceptor is an organic molecule. And because here you do not create an electron or basically a proton gradient in the mitochondria, you do not have this oxidative phosphorylation. So you do not create ATP from ATP synthase. So that's the main differences between cellular respiration and fermentation. So cellular respiration can be both aerobic and anaerobic, and they establish an electrochemical gradient. So this proton gradient. You create this proton gradient that helps ATP synthase start producing ATP. Fermentation doesn't do that. Fermentation basically just uses substrate level phosphorylation to create ATP. So as we spoke with oxidation, so let's talk about oxidation without O2. So this is basically anaerobic respiration and fermentation. So I think we're gonna start with anaerobic respiration. So anaerobic respiration is using inorganic molecules other than oxygen as your final electron acceptor. These can be sulfur, nitrate, carbon dioxide, or inorganic metals. And we'll basically be talking about just small little examples of them. And most of them you see this happening a lot in prokaryotes. So you have some methanogens or sulfur prokaryotes. Methanogens, these guys use carbon dioxide as their final electron acceptor. So basically, these guys will accept electrons and they'll be reduced to methane. Hence, that's why they're called methanogens. And you actually will find these a lot in cows. Actually, this is why cows produce a lot of methane. Actually, their farts are heavy in methane, and this is because they create a lot of methane gas actually through this reduction of CO2 to methane. Then you have sulfur prokaryotes. These are mostly in bacteria, and these get basically guys. They actually take inorganic sulfate, which is SO4, and they reduce it to hydrogen sulfide. And we saw this actually was one of the most like earlier stages of evolution when it comes to photosynthesis. So this is just an example of this Thermoportex tenax. This is a bacteria that actually it loves, sorry, this archaea that actually loves to use sulfate as actually an electron final acceptor. And these guys live in areas where sulfur is very um, rich. So they actually live in hot springs that have a lot of sulfur, which makes sense because they're utilizing whatever there's there. For example, sulfur. They utilize that as their final electron acceptor. So fermentation, we spoke about this briefly, but we go a bit, as a recap. So fermentation basically is literally the using of organic molecules to regenerate NAD. So organic molecules will become your electron acceptors. So this is actually what happens in yeast. So where when you're done with glycolysis, you get pyruvate, right? And you get NADH. But we want to basically recycle it. So what happens is that pyruvate first of all is decarboxylated into acetaldehyde, and then this is reduced into ethanol. And then this helps basically NADH loses the electrons because acetaldehyde is being reduced, so it's gaining electrons, and it's getting electrons from NADH. So then basically then it kind of helps NAD plus to actually be formed. And this is how you create actually alcohol. This is actually literally the chemical equation of creating alcohol. And this is actually very important for people who actually are producing alcohol at home. They use literally this. What they actually will do is get sugars from either of course, from malt, from potatoes, from what other things you could use? I think it's potatoes, malt, anise, different types of, actually, or grapes to create wine. They will use the sugars from these different types of grains or fruits, actually, and they will extract the sugars and they will actually put it with yeast to create actually this whole reaction that at the end gives us ethanol. This actually happens more in animal cells and specifically muscles, and where pyruvate is then fermented into lactate. So pyruvate basically becomes the acceptor and then accepts electrons from your NADH and then to form lactate. And this is basically what gives us actually the this cramps because lactate is then formed in your muscle cells, and this actually prevents a lot of your muscle cells from doing the work. And this is the things that you start feeling this kind of fatigueness. So I'm going to talk about now catabolism of proteins. So here, what I want you to understand is that glucose is the main form of energy. It's one of the main organic molecules that gives us energy, but it's not the only one. We can get energy from proteins. We can get energy from fats, actually, and we can do so. And let me show you first here with proteins. So there's a reaction that's called deamination, in where you actually remove the the basically the nitrogen group. So we know that basically proteins have amino acids, and amino acids have uh, this kind of amino group with this this NH2. If you remove it, you can create actually certain products from the citric acid cycle or glycolysis. For example, here glutamate. If we do this deamination, so the removal of this amino group, glutamate gets converted to alpha ketoglutarate. Alpha ketoglutarate. Where have you heard this? Well, we heard this in the citric cycle, remember? This. So we can actually deaminate amino acids, which then will actually enter the citric acid cycle to give us this NADHs, which again we can then fuel for energy. You know? So it's not only actually, so if you go back here, glutamate that you can do, you can actually do alanine. You can convert alanine into pyruvate. You can do the same thing here with aspartate. You can convert it to oxaloacetate. And this is basically deamination. They do this to deamination is by removing the amino group and converting them into these basically kind of products or basically reagents that will start. So here in this pyruvate, we can actually push the Krebs cycle. Oxaloacetate also can push the Krebs cycle. Another basically source of energy can be fats, and actually fats have much more energy than sugar. They have 20% more energy than basically sugar. For example, if you take right now a six carbon fatty acid, it will yield 20% more energy than six carbon glucose. And this basically what happens here is that the fatty acids are converted into acetyl groups by beta oxidation. So beta oxidation, what it does is basically they can take a fatty acid and break off two of its carbon, basically two of its carbons to create acetyl. And then basically we can then add a CoA to give you acetyl CoA, and that can then re-enter the citric acid cycle. So if you look at fatty acids are long, long chains of carbons, right? So usually these chains are quite even numbered, which is quite great because you need two carbons, so you can keep breaking these fatty acids into multiple acetyl groups. You keep breaking them, and then basically adding CoAs. So you can actually have quite a lot of acetyl CoAs uh, from a fatty acid. So I'll give you an example here. Imagine you have a fatty acid that has 10 carbons. We can break that into five acetyl CoAs because beta oxidation will break the last two carbons always. You'll keep breaking the last two carbons, and we'll keep doing so until you basically it's done. So this is why a six carbon fatty acid can yield more energy because they can actually create much more acetyl CoAs. Because here the six carbon glucose will only produce two acetyl CoAs, as we learned. A six carbon fatty acid actually can give us three. Yes, it can give us three. So that's why it actually can go through one more round of the Krebs cycle. This can go into two rounds of the Krebs cycle because you get to acetyl CoAs, while this gives you three rounds, hence a 20% increase. So catabolism of fat is actually very important. It actually really gives us much more energy than sugars. So this is basically a picture that describes everything what I was saying. You could use amino acids to deamination to let you actually give you products for the citric acid cycle. We can use fatty acids through basically beta oxidation to give us acetyl CoA. We can also use nucleotides. We can use certain nucleotides in where we can actually also do deamination in nucleotides to give us products of the citric acid cycle. So sugars are not the only things that we can actually degrade to give us energy. We can use all the macromolecules. We can use lipids and fats, proteins, and nucleic acids. And as you can see, most of these reactions are um, reversible. So if we create acetyl CoA and we need fatty acids, we can actually then turn it into fatty acids and vice versa. If we basically have pyruvate, we can turn this also into sugars. So now, as the final concept, I want to talk about the evolution of metabolism. So how has metabolism really shifted from the beginning till now? So at the beginning, from the most, let's say, prehistoric organisms, we didn't believe that they had these kind of biochemical pathways. These are very advanced. What we believed is that these prehistorical organisms, how they got energy was by breaking bonds. They were breaking chemical bonds to actually give ATP, to kind of help this substrate level phosphorylation. They were just breaking random bonds to get energy. Then later, we think that organisms developed actually the biochemical pathway of glycolysis. We then believe that they were using organic molecules like glucose to break it down and actually oxidize it fully. And that was it. That's how they were taking the actually energy to create ADP. Then later on, we started to see like, there are certain actually autotrophs that started to come out and started to create their own inorganic molecules, basically from carbon dioxide and light. And however, during those times, there was not oxygen, a lot of oxygen in the atmosphere. So photosynthesis, when you create or organic molecules from photosynthesis, your byproduct actually is oxygen. However, during the first type of photosynthesis, they believed that this was anoxon, anoxygenic, meaning that it didn't produce oxygen. It produced sulfur, actually. And we'll be talking a lot about this actually in chapter 8. And then later on, organisms, specifically plants, learned to actually use water to create basically, instead of this hydrogen sulfide, which was using in the anoxonic photosynthesis. And this basically started to bring oxygen because in this case, when you use H2S to help you to create organic molecules for photosynthesis, you actually release sulfide. If we replace this H2S with H2O, we now basically replace the sulfide with oxygen. And that's what we believe was the change in the Earth's atmosphere. Because before, the Earth's atmosphere was very heavy in sulfur, and we believe because it was these organisms that were doing this anoxygenic photosynthesis. And then later, when they actually switched to this H2O, they used water, oxygen started to come out. And then way later on, we started to feel that it's like, okay, we started to actually take nitrogen from the atmosphere and put it in our metabolism. And this was very important because this started to give us, compounds like amino acids, proteins, nucleoside triphosphates, and nucleic acids that are all need nitrogen. So we started to develop ways to actually incorporate nitrogen. Hence, this is nitrogen fixation. And then finally, the most advanced was aerobic respiration, the one that we talked about going to the Krebs cycle and the electron transport system. This then we think was the most advanced way we basically started to actually develop energy. Thank you so much, guys. This is the end of chapter 7.